All right, let's dive in. Today, we're tackling non-sporing gram-negative anaerobes. And to guide us, we've got Principles and Practice of Clinical Bacteriology, second edition, by Gillespie and Hockey. As medical microbiologists, we know these guys can be tricky, but today we're going deep, getting to that next level of understanding.
I think it's fascinating how these organisms were almost ignored for so long. They were out there causing infections, but kind of flew under the radar.
Hiding in plain sight.
Yeah, it wasn't until we got better at anaerobic culturing around the 70s that we really started to appreciate them.
And their taxonomy, what a mess that was. Trying to classify them just by looking down a microscope. You know, it was like working with half a puzzle.
Totally. Early on, it was all about morphology and a few basic biochemical tests. Lots of confusion, lots of misclassifications.
But then came GLC, gas-liquid chromatography. That changed everything.
Absolutely. Suddenly we could peek into their metabolic pathways, game changer.
Their big reveal.
Yeah.
But the story doesn't end there, right? Chemotaxonomy and 16S rRNA gene sequencing came along and really shook things up.
Oh, for sure. Those techniques gave us a much clearer picture of their relationships, how they evolved, led to some major reclassifications. It's much clearer now how diverse they really are.
So let's meet some of these microbial stars. We've got the big names: Bacteroids, Previtellas, Porphyromonas, Fusobacterium, each with its own quirks.
Bacteroids, now they're interesting, surprisingly aerotolerant, you know? Thriving in the gut, which is mostly anaerobic, but they can handle a little oxygen. They make those mucoid colonies, and lots of them don't move around much.
Like, they've got a secret weapon letting them survive where others can't.
Right. Previtella now, they're a mixed bag. Some have pigment, some don't. And they love breaking down sugars, real saccharolytic, big players in the mouth, and the genitourinary tract.
And then there's Porphyromonas. Slow growers, dark pigments, and, well, let's just say they've got a certain aroma.
Yeah, those volatile sulfur compounds they make definitely give them a distinct smell. Not the best, but it's a clue.
A smelly clue. And who could forget Fusobacterium? Some have that classic spindle shape, but they're diverse, morphologically speaking. They produce butyric and propionic acids, part of their metabolic signature.
Fusobacterium necrophorum is one to watch. A primary pathogen can cause some serious problems like Lemierre's syndrome.
So not just opportunistic, it's actively causing trouble.
Exactly. Then we have Leptotrichia. Looks a bit like Fusobacterium, but it's all about lactic acid production.
Same look, different metabolic agenda. Makes you wonder how they evolved like that.
It does. And Bellophila. Slow-growing, loves bile, can't live without pyruvate. And finally, Bacteroides ureolyticus. Those pitting colonies, strong proteolytic activity, urece positive.
Breaking down proteins, generating ammonia, a real powerhouse.
Indeed. And one thing they all share, they're picky. Need enriched media, Hayman, strictly anaerobic conditions.
We got to create a little anaerobic spa for them.
But yeah.
But speaking of thriving, they're not just pathogens waiting to strike. They're key players in our normal microbiota.
Absolutely. They're part of those diverse communities all over our bodies, contributing to our health, essential for digestion, even training our immune system.
It's a delicate ecosystem. When it's disrupted, that's when we see problems.
Exactly. Balance is key. And that's what we'll be exploring more as we delve deeper into this fascinating world of non-sporing, gram-negative anaerobes.
Let's talk pathogenicity. How do these organisms, often harmless residents, suddenly turn rogue?
Well, it's rarely a single species acting alone. It's more like a shift in the community plus changes in the host. The perfect storm.
The microbial mob, teaming up, exploiting weaknesses.
That's it. And they're armed with these virulence factors, molecular tools for invasion, evading our immune system, causing damage.
Let's break it down. What makes them so formidable?
One of the most fascinating is surface variation. Bacteroides fragilis, for instance, can switch between different capsule types, a master of disguise.
Not just hiding, but actively camouflaging. Microbial spy stuff.
Exactly. And then there's adhesion, their ability to stick to our cells. They use adhesins, like molecular grappling hooks, to latch on.
So they're not just floating around, they're actively targeting us.
Precisely. And some, like Fusobacterium necrophorum, can invade tissues directly, pushing through those mucosal surfaces.
Breaching our defenses, setting up camp.
And they don't always work alone. Polymicrobial synergy. They cooperate, share nutrients, create a supportive environment.
A microbial fortress, protecting each other, making it harder for us to fight back.
And once they're in, they got to evade our defenses. Capsules are crucial here. acting as a shield against phagocytosis. Our immune cells can't engulf them as easily.
Microbial armor.
Right. And Bacteroides fragilis goes a step further. It inhibits macrophage migration. Those are key players in our immune response.
Not just protecting itself, but actively disarming our defenses. A real mastermind.
Indeed. And then there are proteases, enzymes that break down proteins. Porphyromonas gingivalis, for instance, produces gingipains. They degrade parts of our immune system.
So they're saboteurs, disrupting our immune responses.
And of course, there's the damage they cause to tissues. The volatile fatty acids and sulfur compounds from their anaerobic metabolism, those are toxic.
So it's not just their presence, but their byproducts too.
Exactly. They're like little chemical factories churning out toxins, causing inflammation.
And they've got a whole arsenal of enzymes to break down tissues, right?
Okay.
Hyaluronidases, chondroitin sulfatases, heparinases, a demolition crew.
Precisely. Key players in the process. Tissue destruction, abscess formation, they're behind it all.
So they're breaking down our defenses, damaging tissues, causing all sorts of havoc. A multi-pronged attack. But what about systemic effects? Lipopolysaccharide, LPS, triggers a massive inflammatory response, right?
That's right. LPS, a component of their outer membrane, it's a potent activator of our immune system, can lead to sepsis, which is life-threatening.
So it's not just local damage, but the system-wide inflammation that's really dangerous.
Exactly. Interestingly, the LPS of Bacteroides fragilis is different structurally, less potent when it comes to inflammation.
A bit of a paradox. B. fragilis is a common culprit, but its LPS is less inflammatory.
It is. But remember, it's often present in huge numbers in the gut. So even though each molecule is less potent, the sheer quantity...
It's a numbers game. Their individual weapons might be weaker, but collectively they can still overwhelm us.
Precisely. And this difference in LPS, it just highlights their diversity. They've evolved their own unique ways of interacting with our immune system. Fascinating and challenging.
Now let's shift gears. How do these infections typically present in the clinic? What are the red flags that scream, this is anaerobic?
Imagine this, a patient with a necrotizing wound, and the pus has this distinctly foul odor. That's classic anaerobic infection.
The microbial signature scent.
Exactly. And they often pop up in areas with poor blood supply, like deep wounds, abscesses, damaged tissues. They love those oxygen-deprived environments.
So tissue damage plus low oxygen, that's their playground.
Precisely. And the presentation, it varies, depends on the site, the species involved.
All right, let's take a tour of the body. Highlight some key anaerobic infections by location.
Let's start in the gut, their home turf. Bacteroides fragilis is the big player here, often pops up after surgery, trauma, anything that perforates the bowel.
So any breach in the gut wall and B. fragilis is ready to invade.
Exactly. Can lead to peritonitis, abscesses, even sepsis. Serious stuff.
And the female genital tract?
Prevotella is key there. Bacterial vaginosis, pelvic inflammatory disease.
Right, Prevotella bivia, Prevotella decidens.
Yeah.
Those names ring a bell.
They can cause a lot of discomfort. Untreated can even lead to infertility.
And we can't forget the mouth. It's teeming with a mix of anaerobes.
Oh, absolutely. Porphyromonas gingivalis, Prevotella intermedia, Fusobacterium nucleatum, periodontal disease, tooth loss.
And Fusobacterium necroforum, causing Lemire syndrome.
Right. A rare one, but potentially deadly. Starts in the throat, can spread throughout the body.
Amazing how they go from seemingly harmless in our mouths to causing such serious infections.
It is. And they don't stop there. Bacteroides fragilis can also cause bone and joint infections, surprisingly, especially in folks with diabetes or who've had trauma or surgery.
So not just a gut bug, it can travel, cause problems in unexpected places?
Exactly. We also see anaerobic infections in skin and soft tissues, often Prevotella and Porphyromonas.
And those can get pretty nasty, right? Tissue necrosis, abscesses.
Yes, quite destructive. So a diverse range of infections, each with its own challenges. Accurate lab diagnosis is critical.
Absolutely. We need to know who we're dealing with to choose the right treatment. So how do we ID these microbial mischief makers? What are the key things to remember when handling anaerobic specimens?
First and foremost, they're oxygen sensitive. Specimen collection and transport, gotta be careful. Minimize air exposure or they won't survive. We won't get an accurate ID.
So speed and minimizing air exposure. Swabs aren't ideal, are they?
Not really. Pus or aspirates, much better, more representative, less risk of contamination. And we need specialized anaerobic transport media. Keeps those oxygen-free conditions all the way to the lab.
A little anaerobic bubble, protecting those delicate organisms.
Exactly. In the lab, we can look under the microscope, but it's not always clear. They can be small, change shape.
Like finding a needle in a haystack.
Great analogy. But one technique that's helpful is direct gas liquid chromatography, or GLC.
GLC, our old friend. A microbial breathalyzer, right?
Got it. Detects those volatile fatty acids they produce as they metabolize. Even if we don't get a specific ID, it tells us they're there and they're up to something.
A smoke alarm for anaerobic trouble.
That's a good way to put it. Then, of course, there's anaerobic culture, the gold standard. But it takes time, expertise, specialized media, the right conditions. Remember that anaerobic spa?
And some of them are slow growers, right?
Oh, yeah. We might be incubating for days, even a week or more, for those colonies to show up.
Like waiting for a flower to bloom.
Exactly. But once we get some growth, we can start narrowing down the suspects. Morphology, colony characteristics, basic biochemical reactions.
It's like a microbial game of Clue.
And for the definitive ID, we've got a range of tools. Biochemical tests, commercial kits, even molecular methods like PCR or MALDI-TOF.
A whole toolbox of techniques, basic to high-tech.
Right. Which one we choose, it depends. Specifics of the case, the lab's capabilities, how urgent it is.
Speaking of tech, what about those non-culture methods? Are they the future?
They hold a lot of promise. DNA probes, PCR, next-generation sequencing, all being explored, identifying anaerobes directly, no need for culture.
Wow, that's amazing. Fast, accurate diagnosis, no waiting for those slow-growing colonies.
It would be a game changer. But there are still challenges. Sensitivity, for one, needs to be sensitive enough to pick up small amounts of bacterial DNA. Tricky when there are multiple organisms present.
Back to that needle in a haystack.
And contamination.
Yeah.
Got to make sure we're not picking up extraneous DNA, getting false positives.
So accuracy and reliability are key.
Absolutely. But despite these challenges, I think these non-culture methods will be a big part of anaerobic diagnostics in the future. The benefits are too big to ignore.
It's an exciting time to be in microbiology. We've ID'd the mob, analyzed their weapons, now we need to fight back. Treatment time. What are our go-to weapons against these anaerobic foes?
The good news is we've got effective antibiotics. The bad news is resistance is on the rise.
A constant arms race.
You could say that. Judicious antibiotic use and surveillance are more important than ever.
Absolutely. So let's break down the arsenal. What are the key players here?
Metronidazole has been a mainstay. Resistance is still relatively low, broad spectrum, generally effective.
Our trusty silver bullet.
It has been reliable, but we're seeing some resistance emerging, particularly in Bacteroides fragilis.
Not a magic bullet, but still a valuable weapon. What else do we have?
Beta-lactams, like amoxicillin clavulanate or piperacillin tazobactam, are often used, especially for Bacteroides fragilis because of those beta-lactamases.
Right, those enzymes that break down penicillin and other beta-lactams.
Exactly. So we use a beta-lactamase inhibitor to protect the antibiotic.
Like a special ops team, clearing the way for the main force.
Love that. Carbapenems, like imipenem or meropenem, are also effective. Broad spectrum, good against resistant strains.
Our heavy hitters, the last line of defense.
They can be, but we have to use them carefully. Don't want to fuel resistance.
And clindamycin.
Can be effective, but not as broad as metronidazole or that carbapenems. Resistance is a problem, especially with some bacteroids.
Not our first choice, but good to have in the back pocket.
Exactly. And the choice, it always depends, right? The infection site, the suspected organisms, the patient's individual situation.
Microbial chess.
You got it. And sometimes antibiotics aren't enough on their own.
What do you mean?
Well, these infections often involve tissue death, abscesses, surgical debridement, drainage. Those are often essential.
So it's not just killing the bacteria, it's removing the infected tissue, creating a healing environment.
Exactly. And in severe cases like sepsis, supportive care is crucial. Stabilize vital signs, manage that systemic inflammation.
A multi-pronged approach. Antibiotics, surgery, supportive care. These infections are complex.
They are. And as always, prevention is better than cure. So how do we reduce the risk of these infections in the first place?
Great question. What are the key strategies?
Well, most of these are endogenous, right, from the patient's own microbiota. So cross-infection isn't a big worry. The focus is on preventing those conditions that give them a foothold.
Maintaining that delicate balance, preventing the good guys from turning bad.
Exactly. And that often comes down to good surgical practice, especially in areas where anaerobes are abundant, like the abdomen, gynecological surgery, oral facial surgery.
Building a strong defense against invasion.
That's the idea. Minimizing tissue trauma, good blood supply, oxygenation, preventing fluid buildup. Those are all key.
And reducing the bacterial load.
Absolutely. Bowel prep before abdominal surgery, for example. Less bacteria, less chance of infection.
Shrink the enemy's forces before they even attack.
And perioperative prophylactic antibiotics. The goal is to have those antibiotics in the tissues, ready to go at the time of surgery. Stop those bacteria from setting up shop.
A preemptive strike.
Precisely. But timing and duration are crucial. Hit them hard during that critical window, but don't disrupt the normal microbiota or encourage resistance.
A delicate balance indeed. Well, we've covered a lot of ground today. What are the key takeaways for our listeners?
I think the biggest one is this. Non-sporing gram-negative anaerobes, they're more than just opportunistic pathogens. They're part of us, part of our normal microbiota. They play a vital role in keeping us healthy.
Right. Respect the microbes.
And our understanding of these organisms is constantly evolving. New research, new insights into their diversity, their virulence, their interactions with us. It's an exciting field.
So much potential to impact human health.
Absolutely. And finally, I think it's important to remember that it's all connected, the microbial world and our biology. We're not individuals, we're ecosystems. Walking, talking communities of organisms, all working together.
It is a humbling thought that our health depends so much on these trillions of tiny organisms living inside us.
It is. And as we explore this fascinating world, we're going to uncover even more wonders, things that will transform how we think about health and disease. Welcome back to our deep dive into the world of non-sporing gram-negative anaerobes.
Picking up where we left off, those diagnostic and treatment challenges, all thanks to these fascinating and sometimes frustrating bacteria.
It really is incredible how much our understanding has changed, especially when you look at antibiotic susceptibility.
It's like they're always adapting. Those microbial mobs, always one step ahead. Makes you feel like you're running a race you can't win.
You're not wrong. And one of the toughest things we face is how much their susceptibility patterns can vary.
So what worked yesterday might not work today. That's a little unnerving.
It is. We can't rely on those old assumptions, that outdated data. We really need to test each isolate, make sure we're hitting them with the right stuff.
And like we talked about earlier, metronidazole resistance is still low, but it's a concern, right?
Yeah. Definitely a concern. It just highlights how careful we have to be with antibiotics. Can't overuse them, can't misuse them, or we'll just make things worse.
Absolutely. So let's get specific. What are the susceptibility patterns looking like these days? Let's start with bacteroids. What's the situation on the ground?
Well, bacteroids, especially B. fragilis, they're known for producing those beta-lactamases, those enzymes that break down penicillin and other beta-lactam antibiotics.
So they've got a built-in defense mechanism against some of our most common antibiotics.
Exactly. That's why we often need to bring in the big guns, those beta-lactamase inhibitors, clavulanate, tazobactam, that sort of thing.
They essentially shield the antibiotic, right? Prevent those bacterial enzymes from breaking them down.
It's exactly how they work.
It's like a shield wall protecting the troops as they advance.
Perfect analogy. Now, cefoxitin, a cefamycin antibiotic, it used to be a go-to for bacteroids, but we're seeing more and more resistance.
So even our backup plan is starting to crumble.
Unfortunately, yeah. But there's good news. Imipenem, a carbapenem, still works against most bacteroids.
That's good to hear. But like you said, resistance is always a threat.
Always. And we're even starting to see imipenem resistance pop up. Often those metallo-beta-lactamases are involved. Tough ones to deal with.
Those are the ones that can break down even our strongest beta-lactams, right? Like they've got super armor.
That's a great way to put it. It really emphasizes why we need to keep researching, developing new antibiotics. Staying ahead of the game is key.
What about some of the other antibiotics sometimes used for bacteroids? Erythromycin, tetracycline, clindamycin. How do they stack up?
Well, it really depends. Some strains are susceptible, some are resistant. Really highlights the importance of testing each isolate.
So no blanket assumptions when it comes to bacteroids.
Exactly. Individualized treatment, that's the key. Figure out exactly what that bug is susceptible to, then hit it with the right antibiotic.
All right, let's shift gears. Prevotella and Porphyromonas. How do they compare to bacteroids, susceptibility-wise?
Generally, they tend to be a bit more susceptible overall, not quite as well defended.
Not as much armor.
All right, but we still have to be cautious. They can still produce beta-lactamases, so penicillin, some other beta-lactams, they might not work.
So even though they're not the heavy hitters like beta-lactanoids, they can still put up a fight.
That's the thing with these microbes, always got to be on your toes. Amoxicillin clavulanate is usually a good bet. And cefoxitin, imipenem, those also work against most strains.
But again, resistance is always lurking.
Always. erythromycin, tetracycline, clindamycin. We see resistance in Prevotella and Porphyromonas. Testing is essential, no shortcuts.
All right, what about Fusobacterium, Lemire syndrome? That's a nasty one.
It is. Rare, but serious. Starts in the throat, can spread like wildfire. Fusobacterium necrophorum, that's the usual suspect.
And aggressive, too. A forgotten disease making a comeback.
Right. We have to be aware of it, especially with resistance increasing.
So what are our options for treating Fusobacterium?
The good news is most are susceptible to a good range of antibiotics, even penicillin.
So we're not completely outgunned.
Not at all. But there are some exceptions. Ceftriaxone, erythromycin, not so effective.
Well,
And we're seeing more resistance to clindamycin, tetracycline.
so even with the more susceptible ones, we can't let our guard down.
Exactly. Resistance is always a factor. We've got to stay informed, adapt our strategies.
Now let's talk about those non-culture detection methods, the DNA probes, PCR, all that. What's the potential there?
Imagine diagnosing an anaerobic infection quickly, accurately, no waiting for cultures.
That would be a game changer, especially for those really sick patients where every minute counts.
Exactly. That's the promise of these methods. Could revolutionize how we diagnose and manage these infections.
So what are we looking at specifically? What are the technologies?
DNA probes, PCR, those are two of the most promising. DNA probes are like little DNA detectives. They bind to specific sequences, tell us exactly what's there.
Microbial fingerprint analysis.
Love it. And PCR, it amplifies specific DNA sequences. We can detect tiny amounts of bacterial DNA, like a microscopic magnifying glass.
Amazing. Are these being used already?
Mostly in research, for now, identifying anaerobes directly from samples. But before they become routine, we have some hurdles to clear.
Like what? What are the challenges?
Sensitivity is a big one. These methods need to be really good at picking up even tiny amounts of bacterial DNA. Tough, especially in mixed infections. Lots of different bugs present.
A needle in a haystack again.
You got it. And contamination. We have to be super careful, make sure our samples are clean, no extra DNA floating around, or we'll get false positives.
So it all comes down to accuracy, making sure the results are reliable.
Exactly. But despite all that, I think these non-culture methods are the future. The potential benefits, they're just too big to ignore.
It's definitely an exciting time to be in this field. Lots of innovation happening.
I agree. And as we wrap up this part of our dive, I want to emphasize one thing.
What's that?
These non-sporing gram-negative anaerobes, they're diverse, they're fascinating, they're not just pathogens, they're also part of our normal microbiota, playing a vital role in our health.
It's a reminder, right? The microbial world, it's complex. Our relationship with these organisms, it's not so black and white.
Exactly. And as we delve deeper, we keep finding new things, new insights that challenge how we think.
It's a journey of discovery, that's for sure. And I'm glad to be a part of it. Welcome back. Final part of our deep dive into non-sporing gram-negative anaerobes. A lot of ground covered.
It's been quite a journey exploring these organisms. They're so often overlooked, but incredibly important.
We talked taxonomy, their role in health and disease, the whole diagnostic and treatment puzzle, even those new technologies on the horizon. Like peeling back the layers of a microbial onion, more complexity with every layer.
And as we wrap up, I think it's important to step back, look at the big picture. We've focused a lot on the bad, the infections, the defenses they've evolved, but they're also vital parts of our normal microbiota, right? Essential for our health.
Balance. It's all about balance, like a delicate ecosystem, all these microbial communities coexisting. When that balance gets messed up, that's when we see trouble.
Exactly. And this idea of balance, it's super relevant when we talk about the gut microbiota. So many different microorganisms there, including our anaerobic friends.
The gut microbiota, that's a hot topic these days. New research coming out all the time. It's like the gut is the new frontier of medicine or something.
It really is. We're realizing it's not just a passive bystander. It shapes our immune system, influences metabolism, even impacts brain function. It's like this hidden organ, constantly talking to our bodies, influencing our well-being in ways we're just starting to understand.
It's amazing to think these tiny organisms living inside us have such a huge impact on our health.
It really is. And it opens up all sorts of new possibilities in medicine. We're not just fighting pathogens anymore. We're thinking about manipulating the microbiota, preventing and treating diseases, not just infections, but chronic conditions too. Inflammatory bowel disease, obesity, even neurodevelopmental disorders.
A whole new way of thinking about health and disease. A paradigm shift.
Exactly. And research into non-sporing gram-negative anaerobes, it's essential to this shift. Understanding these organisms, how they interact, their effects on us, That's how we'll develop better therapies. Therapies that work with the microbiota, restore that balance, promote health.
So not just wiping out the bad guys, but figuring out how to support the good guys, restoring a healthy ecosystem.
Exactly. A much more holistic approach, recognizing how interconnected everything is. Our bodies, these vast microbial communities, all working together.
So much potential for new therapies. It's exciting to think about.
It is. But we have to remember, we're just at the beginning. The microbiota, it's so complex, always changing. Genetics, diet, lifestyle, environment, so many factors at play. We've barely scratched the surface.
Like we've set off on this expedition into uncharted territory. Every step, new discoveries, new mysteries.
That's what makes it so fascinating, a constant journey of discovery, learning, refining our understanding of this incredibly intricate web of life.
Well said. Any final takeaways for our listeners? What are the key messages you want them to walk away with?
First and foremost, remember that non-sporing gram-negative anaerobes, they're not just pathogens. They're part of our microbiota, essential for our health.
They're part of the team.
Exactly. And our understanding of them is constantly evolving. New research, new insights all the time. Keep learning, keep exploring.
Curiosity is key.
And finally, don't forget that connection between the microbial world and our own biology. We're not just individuals, we're ecosystems. All these organisms working together, a grand collaboration.
A humbling thought. Our health is so closely tied to these trillions of tiny organisms.
It is. And as we keep exploring this amazing world, who knows what we'll find? Discoveries that could change everything, transform how we approach health and disease.
Well, on that note, we'll wrap up our deep dive into the world of non-sporing gram-negative anaerobes. We hope you've enjoyed the journey and that you'll keep exploring the fascinating world of the microbiome. Thanks for listening.